You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.
Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast, and I have Masoud Tavazoi. Should have asked you how to pronounce your last name. I'm sorry. CEO and co-founder of Regenexx, Regenix.com. So, Masoud, thanks for coming. How are you doing?
I'm doing well. Thanks, Rich.
How do you pronounce your last name? I apologize for not asking before.
Oh, no, don't worry. It's a difficult one. It's Tavazoi.
Tavazoi. All right. Well, very good. Tell me about Regenexx or Regenexx. What's the premise of the company?
Yeah, sure, Rich. So Regenexx is a clinical stage cancer therapeutics company, and the focus of the company is to develop what's known as first-in-class cancer therapies or really new cancer therapies to treat patients that otherwise might not have available treatment options for their cancer. And we were founded in 2010 and really based on a scientific premise that we could apply some new technology to search and identify and discover new cancer targets that we could then translate into potential therapeutics for patients. And we've been doing that ever since and now have several drug programs which are in clinical development, which means they're being tested in clinical trials.
So, cancer, as far as I know, there's 100, 200 kinds at least. So which kinds of cancer are you targeting? And, then maybe we'll get into some of the mechanisms.
Absolutely. So we have several different programs targeting different types of cancer, but really scientifically, our approach largely focuses on identifying cancer targets that affect what's known as the tumor microenvironment, which is essentially just a fancy way to say that the therapies we're developing don't necessarily act on the cancer cells themselves, but help act on the patient's body to help facilitate the patient's own attack against cancer. And because of that, the therapies actually have potential to treat multiple different cancers and not just single types of cancer. And that's really the same technology across all of our therapeutic programs. We do focus to some extent on certain cancers where we have evidence to suggest the therapies might work better, and we do so by selecting patients also using biomarkers. So just a little bit more detail for our first program, RGX-104, we're focusing mostly on lung cancer, on two different types of lung cancer there. For our second program, RGX-202, the focus of that program is in what's known as gastrointestinal cancer, so cancers of the gastrointestinal tract, including colorectal cancer and gastric cancer. And for our third program, where it's a bit more broad, it's both solid and what's known as liquid tumors, including leukemias.
So what does cancer do to its microenvironment? What are some of the things that science has observed cancer doing?
Yeah, so this is a really good question, Rich. And right now we're really in a revolution in terms of how we think about cancer, and some of that has to do with technology, but some of that revolution is in our perspective of how we think about cancer. And cancer, as it evolves and grows from kind of a small tumor to spread throughout the body and metastasize and get to late stage, actually evolves multiple mechanisms by which the tumor cells can evade the body's attack on them. Now, the body is equipped to actually mount attacks on cancer, and the body's doing this all the time. That includes the body's immune system targeting the cancer like it would an infection. It's the body's ability to provide nutrients and energy, which the tumor cells can actually take up and steal from the body. It can include simple things like the tumor fooling the body into making blood vessels to feed the tumor, which actually don't come from the tumor cells themselves, but come from the body. So there's multiple different aspects of the body that tumors can actually evolve to manipulate in order to fool the body into blocking its attack on cancer. And so our company has a scientific approach that was developed at the Rockefeller University, which is in New York City, actually, where the company is based, that allows us to model this interaction between the tumor and the microenvironment in animals, in mice, where tumor studies are usually started, to not only figure out how the tumor is communicating with the body, but then to find specific nodes that we can attack with therapeutics that then boost the body's ability to fight the cancer. And most specifically for our lead program, where we have what's known as immunotherapy, which boosts the body's ability to fight tumors using the immune system. And that's a novel approach that other companies are taking, but we found a target on a certain immune cell that can boost the body's ability to fight tumors in a novel way.
So cancer co-ops the cells that are literally adjacent to it or around it, and has them do its bidding, or maybe steals nutrients from them, does all kinds of things to create a local hospitable zone for itself as it grows, right?
That's exactly right. And that's just really begun to be appreciated over the last decade or so. Prior to that, there was a huge focus on making targeted therapies just against the tumor cells themselves, which were taken into a dish in the lab. And we had some success with that, particularly upon the sequencing of the human genome, where we could sequence patients' tumors and look at the DNA and see what is exactly different in the patient's tumor versus the body. But we've become to realize that in itself does not always provide patients with clinical benefit, because tumors can evolve around that. And the interaction between the tumor and the body is really important. And now we have a generation of therapeutics as a community called immune checkpoint inhibitors. These are approved therapies. There's several of them approved, and they actually can provide, in some cases, very long clinical benefit, in some cases curing late-stage patients by training the immune system to attack tumors in a durable way. And so those treatments are not against the cancer like the old generation therapies. They're really treatments to the body that allow the body to mount good, effective anti-tumor response. And so, again, those were discovered by scientists looking at how tumors interact with the microenvironment. And we're doing something similar at Regenexx, but we have a discovery platform that allows us to identify these across multiple different tumor types.
Yeah, I guess the problem is the tumor can affect the microenvironment, in order to sustain itself and grow, but it doesn't send out essentially a systemic response, i.e. it hides from the immune system and doesn't really cause systemic responses. But then when you go to treat it, let's say with chemo or radiation or whatever, that's more of a, you're attacking all the cells in and out of the microenvironment and around it. It has a lot of collateral damage, but yet the tumor is acting in a different way where it doesn't do that except to serve itself. So I can see why immune therapy is one of the ways you could target the tumor locally and without affecting the body possibly negatively systemically.
That's exactly right, Rich. And we came to realize that as a community of scientists and oncologists by really studying what we call in vivo interactions between the tumor and the host, and not neglecting the fact that that interaction is actually very important, and that's how tumors grow. Initially when we started looking at cancer again 2 or 3 decades ago, we didn't have some of the tools and techniques to do that. So we just take tumors in a dish, see what made them grow, and then find basically toxins against them, which, as you said, can affect the body in detrimental ways. But now, as we have good, very strong scientific and biological understanding and have some technology that we can apply, we can then really tease out the interaction. And we've learned that tumors evolve And they actually, the body has a lot of mechanisms to fight tumors, but these tumors can kind of turn those off one after the other in what's kind of commonly phrased in immunotherapy as checkpoints. They turn these checkpoints off, and then the body thinks, oh, we should mount an immune response to this tumor because the body doesn't recognize it as a tumor. Those mechanisms that the tumor is taking advantage of are mechanisms that the body uses to protect itself sometimes for an infection, for example, where you don't want the body fighting a cold for 10 years. You fight the cold for a week and you recover, and then you turn the immune system off. Tumors evolve to actually learn how that happens. It's really interesting. They're very clever because they can evolve and through a population of cells to kind of turn on and off cellular pathways, and they arrive at this. And so we just model that in animals and look at the kind of whole multitude of proteins that can change and find those changes that really are affecting how the body interacts with the tumors. And then our mission at Regenexx is to make therapeutics against those. And I'll just say one more thing, Rich. An advantage of that is not only that you can really affect those key nodes and hopefully get durable benefit, but there's the potential to really go broadly across tumor types, because in many instances, across different tumor subsets, the same mechanisms are being applied, whether it's a cancer maybe in the lung or a cancer in the skin. So this knowledge base that we're accumulating as a field and Regenexx in particular for our therapeutic programs could have potential therapeutic benefit across multiple tumor types.
Yeah, it's weird. You have a primary tumor. It lives, let's say it starts in the liver. So that's a unique environment, and it creates its own microenvironment to thrive. Then it grows and metastasizes, let's say, the pancreas. Completely different environment, yet it still survives there, and it still manages to take hold and grow and form a niche, and then maybe it metastasizes to a different tissue. How does it do that? How can it seems to go in and sense and then alter itself to be able to thrive in multiple different environments that could be radically different from where it started? Is there any learnings there that we found?
You're absolutely hit on the right point. And at the basis for our technology is exactly that. We start with human cancer cells from patients with known cancers, and we model metastasis in animals. And what's interesting is the cancer cells in mice actually metastasize and behave and travel to the same organs that same cancer would in a patient. And then we look at those cancer cells after they've evolved, grown, and become very aggressive, and we can compare them to the original tumor, which you can kind of think of as a primary tumor. And then we say, what's different between the cancer cells that made it and those that didn't? A good analogy is like if you want to swim across the English Channel and you're looking at your country's athletes, all the top swimmers, you could choose 2 or 3 of them, or you could say, I'm going to take 1 million people and have them all try and swim and see who the best swimmer is. You're going to arrive at a couple incredible swimmers that can make it across, and then you go look at those people who made it across and you see that they have a swimmer phenotype. Cancer cells do the same thing. They start small, but they evolve. As a population, they undergo changes, and some cells have better adapt to live in the liver. Some are better adapt to live in the lungs. And while the population evolves, they effectively shut down the body's ability to control their spread. And they do this using all the different various things we spoke about: immune system, energy, blood vessels, other cells. And that's how they almost evolve individually as cancer cells, but then grow as a population. And depending on which organ they go to and what type of cancer, they utilize slightly different techniques, and we can learn from that by modeling that. And that's exactly what we do at Regenexx, and that's where the technology is. We really model that to mimic what what happens in cancer patients who have advanced metastatic cancer, and then we can learn from that and find novel targets.
Have you tried, let's say, liver cancer in a mouse model and taken cancer cells and put them into the mouse's liver? And does that cancer always tend to go to the same metastasis sites, or is there any signal, or is it just random where it goes? And then what if you took that same cancer and you put it into the pancreas? Would it leave the pancreas and go to the liver and then metastasize from there, or would it have a different path? Have you tried things like that?
It's pretty interesting, Rich. If you take from a patient from their blood metastatic cells, for example, from a breast cancer patient who has metastasis to their lung, meaning their cancer spread to the lung. If you inject those cells into the venous bloodstream of mice, they tend to go and metastasize to the lung. Now, they might metastasize to other areas. It's not an exact replicate of the biology, but there's a surprisingly consistent migration of certain cells, and that's because certain tumors that grow certain places, at some stage, they're adept to migrate to certain areas of the body. And in many cases, a cancer that maybe has spread to the lung might have also spread to other areas of the body, but there might be a survival advantage in the lung, where that's where you end up seeing the tumors, whereas in other places, maybe they have a small amount of cancer there. And again, we have to learn from these biological processes, because it suggests that something of that cancer has been able to take advantage of the microenvironment in the lung, for example, and then we want to learn what that is. Why is it that they don't grow so well early on in the lung, but eventually they do? What's the difference between the cancer that can grow there and the cancer that doesn't? Because that can reveal, through kind of subtractive analysis, the key things that the cancer needs to grow somewhere. And that's why we care about. We want to treat patients. We want to have a patient-centric model of how we think about cancer. And each patient is different, and each cancer is slightly different, and how the cancer relates to the body is different. And so we want to learn from that and really think about the way you pose the question is a good one, because using that subtractive analysis, really, and comparative analysis is the best way to describe it. You can then see, all right, well, these cells that grow here, they're using this protein to do so. This protein, as an example, gets secreted, and that shuts off the immune system. Maybe that's not so important in certain parts of the body where the immune response is not very strong to begin with. And so that's not important there. And so we can use this subtractive analysis to really find, the key nodes that these cells use. And I'll just say one other thing, because I think it's important as we think about therapies. Often you have to hit multiple different nodes at the same time to really get good benefit for patients. And we're seeing that in clinical trials, where some of the best therapeutic approaches now in this kind of next generation of immunotherapies are by combining certain immunotherapies with other therapies, even including chemotherapy, which can help boost the activity of the immune system in ways that we're just learning after using chemotherapy for 30 years. So it gets very complicated, but we can simplify it by using technologies that really get to the key differences of what makes metastatic cancer so aggressive versus maybe a cancer cell type that's not able to grow there.
Yeah, I was thinking about the breast cancer example, that it tends more often than not to spread to the lung. I guess that suggests two things to me. One is that maybe when the breast cancer is ready to metastasize, just the way it changes, mutates, predisposes it to be able to function in the lung over other organs. That's one idea. The other idea is, what if the cancer is scouting for other locations and it finds the lung, and then it communicates back with the primary tumor, the cells that are in the lung, and says, all right, here's what to do, and you've reached a location, now we'll send more reinforcements and have you build up there. Just two ideas, pure speculation, but have you seen any evidence of either of those things?
Yeah, there's multiple different ways that this happens. In some cases, the anatomy actually can drive to some extent where cancers go. So, for example, colorectal cancer and pancreatic cancer tend to spread to the liver, and that has to some extent to do with the way that the anatomy is of the liver, which drains a lot of the blood that comes from the gut, and that's how we digest food and get nutrients. And in other cases, for example, for lung metastasis, the lung is the area where blood goes through to provide oxygen, provides an easy conduit with a very wide surface area. But what you said is interesting, because cells that metastasize and spread often send signals to the entire body, and we know this because those cells can actually globally suppress the immune system. Metastatic cancer in one part of the body can send signals to the bone marrow, where immune cells are made, to actually generate immunosuppressive cells that usually are only present when the body wants to shut off the immune system. But the body's immune system is tricked by the cancer to do that. So it's very interesting, because you now have a system where the cancer is interacting with the body in a detrimental way. And once we figure out what that signal is, and in our Regenexx, for our lead program, we've identified what we think is one of those signals and have a therapeutic compound that we're testing on that premise, you can then shut that signal off and block the ability of those aggressive tumor cells to manipulate the body's immune system so that you don't get more spread. So this is part of the appreciation of the complexity of cancer, and it's a good thing because we then start to think about cancer as a systemic disease and not just a tumor in isolation, because the body is very important and its interaction with tumors are very important. A patient's health status is very important. A patient's immunologic status is very important. All these things we're now coming to appreciate, and we're applying technologies to take advantage of that, because the goal is to really cure patients of their cancer, right? And we're starting to see that in some solid tumors with immunotherapies, but the problem is we still, despite these advances, the majority of patients who have advanced, stage 4 cancer, for most indications, will succumb to their disease. So we have a long way to go, but this revolution, I think, in how we are approaching cancer is gonna really help identify these kind of complex mechanisms and distill them to the most important ones that we can then attack with novel therapies.
And you also said, you alluded to the fact that in order to treat cancer, you need to essentially attack it on multiple fronts, because it seems to be able to hyper evolve with incredible speed. And I've heard of people getting chemotherapy of one type. The cancer grows smaller, but then it comes back stronger and more aggressive than before. Then they hit it with radiation, same thing. Then they hit it with another type of chemo, and it develops resistance against multiple attacks that are serial, but I guess in parallel is how you could really hopefully get rid of a cancer.
That's right, and some of these older generation chemotherapies actually do provide good benefit for patients. And what we're learning now is that in some cases, instead of providing patients sequential therapies, for example, with chemotherapy, sometimes combining these at once with immunotherapies in one combination regimen actually is better than providing each one individually. And again, we're learning now that even things like chemotherapy that we thought maybe just affects cancer cells might actually provide the bodies a better ability to mount an immune response against the tumor. So there's all this new learnings that we have as a community working on cancer medicines that's kind of re-looking at the past therapeutics, and then the challenge becomes, which is that we have these novel therapeutic programs that are not approved, obviously, that we're testing in clinical trials. We then need to see, do these therapies that are approved work well with the ones that we're currently investigating. So then you have the added complexity of doing combination studies. And this is where really understanding the biology of what cancer does and understanding the biology of the novel targets that you're making a therapeutic agent against really are important, because then you can think about ways to rationally combine different cancer modalities, whether it's chemotherapy with immunotherapy or radiotherapy with immunotherapy, or two different types of immunotherapy hitting two different types of immune cells. These are all key elements of cancer drug development nowadays, and it's one of the challenges that everybody's facing, really, of how to appropriately combine agents. And so at the same time, it could provide a big benefit as we see some potential synergies in combination therapies. And again, we're seeing that in some instances. For example, in lung cancer, non-small cell lung cancer, as a first-line therapy, the administration of these checkpoint inhibitors with chemotherapy is providing pretty good patient benefit. And so there's obviously a lot of room to improve, and a lot of patients still don't have effective therapies, but the biology is important to understand the biology.
What about cancer cells themselves? I've heard there's cancer stem cells and there's regular cancer cells. I mean, the cells that, let's say, are responsible for metastasis, are they of a third flavor or type? Have we looked at that kind of thing?
Yeah, it's a good question. A lot of this has to come down to a little bit, nomenclature. What maybe some scientists might say are the cancer stem cells might be the similar types of cells that are metastasis initiating, or some of those cells that are actually resistant to therapies might also be those. So the way we look at the biology, we try and simplify it. We don't necessarily like to put labels on the cells. We like to look functionally at what happens because we really just care functionally. Can we really block cancer progression? Can we block the metastasis of a cell of cancer cells in totality? So we actually look at entire populations of cancer cells and do analysis of their of the signal that they send out to the body. And we don't like to take necessarily an approach where we have to know ahead of time which cell is important. We let the biology tell us by modeling cancer spread and metastasis in animals, and then really just comparing the tumors in totality. And then that also means we have to look at the immune response in totality as well, and look at other things like energy, metabolites in the body, and things like that. So we try and, again, at Regenexx and our scientific team to try and get away a little bit from the phenomenology or the names and look kind of functionally, which is obviously a challenge. But some of those same cell types you mentioned are overlapping, and that all comes about by cellular evolution of these cells, which is the other interesting phenomenon, which is a tumor at one point might look very different a week from now, and a metastasis might actually have very different cells in one place than a metastatic tumor in another place, and might have different interactions with the body. And these are all things that we need to really understand at a deeper level as we work to make more effective therapeutics.
Yeah, have there been 3D images of a tumor to look at the structure of it, the types of, you talk about it in terms of function, but the function of the cells within a tumor, do they appear to come in layers of function, or is the tumor differentiated in its function, for instance?
Yeah, there's more and more technology being put into looking at even single cells, isolating them from tumors, characterizing the expression of the genome in one cancer cell versus another, and then doing the same thing for all the immune cells that go into a tumor, and realizing there's different types of T cells in a tumor, there's different types of myeloid cells in the tumor, different types of there's even B cells in tumor. So there is a push to really fundamentally characterize what's the heterogeneity of cancer, and then the heterogeneity of I'm using the immune system as an example because it's the best example we have of a tumor microenvironment target, but looking at the heterogeneity there and really understanding that it's a dynamic process. It changes over time. At one point, you might have the immune system mounting a powerful attack. At another point, you might have the immune system be suppressed by the tumor, and that in some cases can be reversible with immunotherapy. So we're having to build not only models, but we have to use technology and really have access to samples from cancer patients, which are being volunteering in clinical trials for therapeutics, and these are all building our data sets and building our experience of how cancer works. It's really very important as we move these potential therapeutic programs forward that we have a good understanding of the heterogeneity, and that's something that the field as a whole is working very hard to characterize.
It's so complicated. I mean, how do you direct the company's work? How does the company know what to do? Because it's just amazing. I mean, cancer is like its own organism. It's like a parasite. It has its own goals. It wants to preserve itself. It's able to spread and proliferate and adapt, and it's just unbelievable.
It seems pretty daunting. Obviously, the community as a whole builds on the science, but fundamentally, when we started this process, we again wanted to just distill it and kind of simplify the way we think about it. And so we just started with this premise that we're going to start with some cancer cells that we isolate. We're going to characterize what proteins they express, and proteins are the outcome of the DNA. That the proteins are what makes a cell, and that's what provides signals to other cells. And then we're going to model how cancer metastasizes in humans, in animals, in the lab, and then we're going to generate highly aggressive cancer cells from the population of cancer cells we started off with, which are from the same patient, are genetically very similar, but behave very different. And then we just literally do the comparison and say, what's different about these highly aggressive cancer cells that have metastasized to the lung or the liver, and how are they different from that first set of cells that we started with that could spread but didn't do so aggressively. We really do a selection process, and then we just look at the profile of those cells molecularly, and a lot of different things come up. And then we focus on ones that really seem to be happening in more than one patient's cancer cells. So then we can do this broadly and say, all right, well, specific change is coming up very consistently every time we look at this metastatic process. This must be important. What is this doing? And then we can use genetics and molecular biology in the laboratory to tweak those genes and the proteins they make. And then we say, oh, aha, this actually drives cancer progression. So we really start off what we like to say is an unbiased approach. We pretend like we don't know any of the proteins that are important. We just know the fundamental process of cancer spreading is important because that's unfortunately what causes most cancer mortality. So that's what we want to study. And then we model it, and then we let the biology come back to us and say, well, this target is really important. It seems to be coming up every time you have a cancer spread to the lung. And then we say, all right, well, maybe that's a good target. And then we work to make therapeutics, and that's really what the company does. We do drug development and move programs from early science all the way to clinical trials with potential agents. So obviously it's a complicated process, but biologically we try and simplify it that way, and that's possibly why we've been successful at getting novel compounds into clinical development based on the science.
Have you tried to see the very inception of cancer? Have you let's say injected a mouse with one cancer cell or just a few and try to tag them and track their progress? Or have you ever observed the very start of a metastasis, and would that give you any insight?
Yeah, so Rich, what happens is if you take a population of cancer cells as the first time and you inject them into mice and look for cancer spread, you need several million cells usually just to even start. And after many months, if you can label them with a protein that a detector can detect that fluoresces outside of the mouse's body. It's pretty interesting. You can do fluorescent labeling of cells. You can monitor them. After many months, you get small tumors in the lung. Now, if you then isolate those tumors that have spread to the lung and re-inject them in another round of animals to model it, and you do that many times, at the end of it, you can inject maybe just a few thousand cells, and you can get very, very aggressive tumors to grow. You started with having to inject very many million, and that's because we're starting to isolate the highly aggressive population of cancer cells that provides metastasis from the first population that was more heterogeneous. And so we can monitor even very small metastases that start to grow and look and see what happens there. And it's pretty interesting because the cancer actually starts forming a niche the second it arrives in an organ. It starts to affect the way the blood vessels are. It brings in a handful of immune cells that are immunosuppressive, so T cells don't come and attack it. It starts to modify the energy metabolites around it. So even from an early, very, very small metastatic tumor, those tumors are already evolving to grow into their niche. And this is the challenge with cancer, right? We often diagnose cancer at a very late stage and at advanced stages, which is already metastasized. So the barrier for really targeting those cancers is high. And so we have to really try and find these effective therapies that hit multiple different aspects of cancer growth, metastasis, spread, because it's not just about metastasis. Once the tumor finds a site, it grows and brings in other nutrients. So our approach is not just geared towards metastasis, it's really geared towards finding those key elements that make cancers grow kind of all through the body, and the really key, Achilles heels, as they say, of the process, and then figuring out how to combine them effectively.
I know this would probably be a really horrible experiment, but has anyone infected mice with cancer and then bred them and see if they go through to the germline and are inherited and expressed in the
generally, I don't know of those experiments. We don't do them, certainly. I think cancer basically is pretty localized to the one generation. Yeah.
Well, thank God.
We're aware of mutations. Yeah. Obviously, Rich, we know of certain mutations that we have as humans that render us more susceptible to getting certain types of cancers. And those, obviously, as you know, are inherited. And there's examples of that, for example, for breast cancer and ovarian cancer. And those are genes that predispose to getting higher rates of cancer. And so that's an inheritable form of cancers, actually, that predisposes people. And there's been a big push in the field to try and not only diagnose patients who have those early, but then make therapies against those specific targets. And the field is making some progress there. But again, there's a lot of work to be done because a lot of patients still don't have effective therapeutics. So that's really our mission, is to push this progress forward, and because a lot of patients are still in need. And despite the big advances we made in technology, we have a lot of work to do as a community. So,
I know.
The challenges remain.
Well, I guess one more question referring back. The reason why I asked you if a single cell could cause cancer, let's say in a mouse model, is that, I've heard around from many scientists, cancer can start from a single cell. But do you think it does? Or do you think it really takes a whole collection of, let's say, several thousand cells in a given area in order for it to start? Because why would it take so many in a mouse to start, let's say a non-aggressive form of cancer? Why wouldn't one work?
The way that I look at cancer is really, it's an interplay between what could be a really small number of cancer cells and the body's ability to defend itself against those. And that's happening all the time. There might be, unbeknownst to us, very small number of cells that aren't tumors, but are starting to get the characteristics of what a cancer cell would look like. And maybe early on, before those cells have had a chance to become too aggressive, the body can actually mount an attack against those and eliminate them from the body. That happens all the time. And that's happening in all of our bodies, right, to some extent. At some point, there's a tipping point, really, where the body then loses the ability through the tumor itself modifying and growing very aggressively to not be able to sustain the type of attack it needs to on the cancer. And that's when we, start having to need to deliver therapeutics. And so that way I think about it is the balance between the attack of the tumor, and then for immunology we think about it as immune activation versus immune suppression, and the interplay between that and the tumor. So I like to think about it like that, because that seems to me like what the data suggests is the case. And I don't like to think about so much maybe quantitatively how many cells does it take, because it's dynamic, and what that suggests is that there's multiple things that have to happen for a cancer cell to become a very large tumor. It doesn't just grow quickly overnight and grow. There's some evolution of the tumor, and then there's some effects very likely that are being mediated into the body itself that allow that tumor to grow without the body being able to attack it. And that's where I think we're making a lot of progress in our understanding of the biology. And from our perspective, I think that's really this revolution that we're in now with immunotherapies and other therapies that affect the tumor microenvironments, to really understand that, because I think that's where you're going to get the best benefit for patients, understanding that and then targeting that. So you're facilitating the body in a way that the body has already learned to fight early and small, potentially cancer cells and tumors. You're just boosting the body's ability to do that, and unraveling or eliminating the veil that the cancer puts on itself by evolving all the time. That's the way we look at it, and that seems to be becoming a prevailing kind of perspective on how cancer behaves.
Well, maybe by studying a particular kind of cancer that acts in a more predictable way, that you can learn its playbook and the order of what it does and how it does it, how it becomes, it goes from stage 1 through 4, let's say. Maybe that would shed light on, how to counteract it at different stages. If you learned, again, like I said, a certain cancer, a predictable one's playbook.
That's right. And as a community, there's certain cancers we've done that. For example, colorectal cancer, because of the advent of colonoscopy, we're now, and to some extent, it's a preventable cancer. We can see, early polyps growing in the colon, and those can be removed and analyzed, and that work over time and the science behind that has revealed that that cancer is a kind of for that example, but it probably holds for other examples. It's kind of a multi-stage process, and it starts one way and it evolves, and at least for that cancer, because of colonoscopy, you can prevent it by removing early stage polyps that might actually have turned into cancer later stage. And that's actually taught us a lot about the kinds of mutations that happen in cancer early, and then how cancer grows beyond the colon and spreads to the liver. That's all helped the field of oncology, and at the same time provided a valuable note to prevent the onset of late stage cancer. But unfortunately, we don't have that for most cancer types because they're not readily accessible. We can't always monitor like we can in the colon with colonoscopies. There's obviously mammograms and other preventative measures which are very valuable, but for a lot of cancers we can't monitor globally the body on a daily basis. So we have to come with a mission to really treat aggressive stage 4 cancer, which is what we often see in the field. And those patients really need therapies because they're very sick. And so we have to kind of look at it from that angle, but at the same time learn from earlier stage cancers that we've had a growing body of knowledge about from the oncology field on how they spread and grow and the science behind it.
But again, like let's say for colorectal cancer, is there a well-trodden path that particular cancer tends to take with defined steps that have been observed to find changes in it?
Yes, in many cases, that's been pretty well studied, and that's one of the better models we have. And a lot of that, again, is from the ability to access, polyps and early tumors by colonoscopy and resection. And there's a pretty consistent pattern of, a subset of cells overgrowing, becoming what's a polyp, and then what might look more like an adenoma, and then, these polyps getting larger and larger, and then at some point, potentially due to mutations in their DNA or other events, becoming really malignant cancers, which then by definition spread beyond the colon and usually, not always, but usually will then spread to the liver and grow in the liver, and then after that grow to other parts of the body. And even just the presence of multiple tumors in the liver is a sign that the tumor is more aggressive and doesn't respond, and those might not respond so well to therapy as an earlier stage tumor does. So that process is characterized. Obviously, we don't know all the molecular steps. That's the second program at Regenexx that's in clinical trials right now is geared towards gastrointestinal cancer, and it's really based on that the biology of how colorectal cancer spreads. That we've gotten some insights into potential ways of targeting that with our second program that's in clinical development, RGX-202, based on the biology of colorectal cancer progression that we've uncovered using our scientific approach.
Okay. Well, very good. So you said you're in clinical trials. What is the near-term future? And then, maybe the next year or two, and then the next 5+ years, what does it look like for Regenexx? What do you think will be coming?
Sure. So we have a lead a compound. It's called RGX-104. We completed what's known as a phase 1A dose escalation. That's essentially mostly a safety study, but we also were able to monitor the immune activation and anti-tumor activity there. We're starting in the summertime a phase 1B/2, so that's mid-clinical development for that program, geared mostly towards lung cancer. Our second program, RGX-202, as I mentioned, is in a phase 1A for gastrointestinal cancers. Again, that's the first stage of clinical trials. It's a small molecule. Again, these RGX-104 and RGX-202 are what's known as small molecules, meaning they're delivered orally. They're not injectables. And then we have a third program that has not entered clinical development yet, that will enter clinical development roughly in about 18 months from now. So that's what we call preclinical. And then we have our target discovery approach that identified the targets of these three programs that we're now applying to find even further novel targets. So we're really built the company here in New York to be not just a developer of specific programs, but really an all-encompassing company that has a target discovery platform to continue to do the science. And so the next year for us is going to be an exciting one as we take RGX-104, our lead program, into this phase 1b/2 studies in lung cancer. Those are going to be pretty large clinical trials across the United States, and our second program as well through the clinic. So we're going to be getting interim data into 2020 for both of those programs in those select tumor types, and then bringing our third program forward. At the same time, we at the end of last year closed our Series C financing of $40 million. We've raised more than $80 million to date, and so the company is well financed through 2020. So we'll be looking again to kind of push all the programs forward and really build out the company as really an independent novel cancer therapeutics company to really make these therapies for high unmet need tumor types for patients that don't have effective therapies.
No, that's great. I guess last question in general, how effective do you think it'll be? Your efforts and other companies, do you think that will, be able to at least extend the life significantly of people with various cancers in the next few years, or is it still a long road ahead?
Yeah, that's our goal. Our mission is really to, extend the life of patients with cancer and ultimately have patients that have advanced cancer be cured of their cancer. And as a community, we're starting to see those types of responses with immunotherapies that are already approved. The checkpoint inhibitors is the example I gave, and there's several of those approved both in the United States and Europe and globally that have provided long-term, more than 10-year clinical benefit from early studies already in advanced cancer patients. And for all intents and purposes, some of those patients are cured of their advanced cancer, and that's really the first time that's happened. And that's really emerged over the last, 5 to 10 years. We've been building on that immunotherapy progress. And so, again, that's a small percentage of patients get that benefit. A lot of patients don't get benefit from those therapies, and we're trying to figure out why as a cancer community and overcome that. But we're really in a phase, I believe, of a lot of growth in the knowledge, science, and technology that will have a larger and larger number of patients getting the types of benefit that we all really dream of, which is really long-term cures and not just, a few weeks of clinical benefit from therapies. Because I think that's what patients are in need of that. So as a community working on this problem, we have to really focus on the big need. And so I believe that that's coming. Obviously, it takes a very long time to develop a therapeutic. You have to start with the basic science of many years, but a lot of that science has been going on over the last decade, particularly in immunotherapy, and we're starting to, as a community, again, reap the benefits of that. I think with the approval of checkpoint immunotherapies, and then these kind of second generation immunotherapies like the ones Regenexx is developing.
Well, very good. Well, Masoud, what's the best way for people to contact Regenexx, ask questions, suggest collaboration, et cetera? Yeah,
so we have a website. It's at www.regenexx.com, RGENIX, and there's a portal there that they can send comments to and any inquiries.
Okay, well, very good. Well, I appreciate you coming on the podcast.
Thank you so much, Rich. I appreciate it.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.